So what I want to talk about basically is following on from what Jared already described. So he gave a very nice introduction to the idea of DIA, and I just want to spend a bit more time talking about the details of targeted extraction and a little bit untargeted extraction, which already came up in the discussion of the last lecture. But there won't be too much detail on this, but just to say that it basically exists. So where are we in the context of the course? So Jared just gave this nice introduction about the DIA concept and a little bit about targeted analysis. So by going from DIA type data and doing an extracted ion chromatogram analysis, you can get to something that looks kind of like SRM data. And earlier in the day, Ludovic talked about mProfit and automated peak picking and error rate control, mostly in the SRM context, but the idea here is if you have something that looks like SRM data, you can do an automated analysis and you can get automated peak picking, you can get estimates of the false discovery rate and so on. And so the idea is here whether we can put these two kind of concepts together so that now we can go to essentially automated analysis, large-scale data, automated large-scale data analysis of SWATH or DIA data. My slides are a little bit cut here. I'm not really sure why that is, but just keep going. So the outline is make a few brief comments in terms of introduction about historical perspective and kind of maturity of targeted approaches, and also quickly mention about what other softwares are available. I'll quickly go over the targeted analysis concept that Jared just described, but more from the kind of direction that we tend to think about it around here and the original SWATH paper. I'll talk a bit about this software OpenSWATH, the implementation and this kind of proof of concept surrounding OpenSWATH. And then I'll mention a couple of, well, 3 different applications actually. And the point of the applications is not really to show you some biological applications. We'll look at these in a very cursory way, but more what I want to point out is that this kind of works at different scales. And then I'll very quickly say something about untargeted analysis, which we already kind of got into the discussion after Jared's talk. So here's Tina's kind of nomenclature slide again. So now we're down here in the SWATH DIA world. Jared used mainly this term data independent acquisition. I will probably say SWATH more than this. To me, they mean something slightly different. Here's a slide from a review which I like, and this kind of shows a historical perspective, a historical overview of the development of data independent acquisition. So what I want to kind of point out with this is that there are many different flavors of DIA, and we kind of regard this SWATH idea or targeted analysis of windowed DIA data. This is what we referred to as SWATH in this paper a couple of years ago, whereas data independent acquisition encapsulates many things, right? So this started back with the concept, this was a paper from Dick Smith, it wasn't really DIA, but the idea that you could do multiplexed MSMS. So you could co-isolate 2 peptides and fragment them together and then somehow interpret the spectrum. Later there was a paper from Dave Goodlid where he called shotgun in-source CID, and here they basically fragmented everything. It kind of looked a little bit like the modern implementation of MSC. The paper many people are familiar with is this one from John Yates in 2004, where they kind of introduced this term DIA, data independent acquisition, and also introduced this idea of kind of windowed acquisition. So slicing up the precursor master charge range in windows of 10 Daltons or 20 Daltons or something like this. This was the first implementation. MSE was this commercial implementation where they fragment everything at the same time. And there have been, well, a bunch of others, including Pacific and AIF. This is the paper that came out of our lab in 2012, which Ludovic Gillet wrote. And essentially, this was kind of, at least for us, was a turning point. And the really thing that made the difference was the use of targeted data analysis, which you've now heard about several times. And this, in our hands, is kind of what started to make things work. And since then, what we've been working on is, enabling software tools, software, and assay libraries, and various different applications. So if you're interested in this kind of historical perspective, I think this is a nice review from Dave Goodlid and some others. So I quickly did a couple of days ago a search of some terms that I thought represented selected reaction monitoring and multiple reaction monitoring, or SWATH and DIA, what the number of citation or what the number of articles is, for these various methods, right? So you see in the last 10 years or so that SRM and MRM for proteomics has taken off in a major way. There are a very large number of publications, and I think if people believe it's a very credible technique for quantitative proteomics now, and this is what we've heard all week. It's much less mature for SWATH and DIA, and this is kind of what I want to point out with this slide, is that we're really at a closer to the beginning now, and this impacts a little bit how we do the analysis. So we're much more in a development phase, I think, in the SWATH and DIA world than we are in the SRM and MRM world. Well, I zoomed in here just to show that it really is going up, right? So there's some kind of exponential growth here, but it looks a bit sad down here, so I had to make an inset. And this kind of is encapsulated here as well. If you do kind of what I will call a classical proteomics, targeted proteomics experiment like SRM or MRM, or now increasingly PRM, and you want to do the data analysis, well then, I mean, Skyline is basically the one-stop shop for this. You can do essentially everything you need to do within Skyline. In fact, Skyline has had a really significant impact on the field of targeted proteomics, but so this is a done deal, I would say. For kind of newer generations of SWATH and DIA, I would say it's a little bit more up in the air. And there's many tools available. In conferences the last 1-2 years, people are presenting very different ideas and ways to do this. Some of them look similar to each other. Some of them are are different. DIA Empire, for example, we mentioned. But I mean, what I'm trying to point out here is if we take, this is a work in progress, but if we look at a list of some of the available softwares, this is not even all of the available softwares, and maybe we split them up into kind of targeted and untargeted categories, there's already a large number of tools available in both categories. The one I will mention mostly is this OpenSWATH tool that was developed here in Zurich. I want to point out that the implementation is that's now in Skyline is rather similar to what I will describe in OpenSWATH. So what I say about OpenSWATH will be kind of relevant to Skyline as well. And then right at the end, I'll just make a few comments about this DIA umpire that was already mentioned in the previous talk, just to kind of point out to you that there are things happening in the untargeted kind of world. And then Lucas Reiter will be here, I think tomorrow maybe, and he's going to talk, part of his talk will focus a little bit on the Spectronaut software. So this is a small company in Zurich, and they've also developed a commercial platform for doing this. He should say something about this, I think. In terms of the targeted software, most of them, from a high-level conceptual way, I think they work fairly similar. So Ludovic pointed out that peak view is maybe a little bit different in terms of the way the decoys are estimated and the false discovery rates and so on. But these guys up in the top half are more or less in a high-level conceptual way do the same thing. You extract ion chromatograms from DIA data and you try to score it some way versus some decoys. The untargeted way is a bit more diverse and also kind of much more under development, and I won't say much about this except this small example of DA. So just to remind you kind of why targeted analysis, somebody asked, can we look at a spectrum that comes, so a SWATH or a DIA MS2 spectrum. So here's an example of one. And you can see it's very busy, right? And just to point out that, I mean, what's the problem here? SWATH MS-MS spectrum contains fragmentines from any peptides which are in this precursor isolation range. In this case, it's 600 to 625. Any peptides that are currently eluting. So this spectrum is a composite of fragmentine spectra from whatever peptides are in this precursor range. And this is actually from Skyline. Skyline has highlighted a few of the fragmentines for a particular peptide, which we have a library spectrum for. But you can see that these are the major fragmentines of this peptide, and they're kind of buried down in the noise here. So you have noticed somebody asked, is there a chance to search directly a spectrum like this in Mascot? And the answer is, well, directly not really, because all these other major fragmentines will dominate the way this spectrum is scored. So to remind you, we need this is a targeted analysis approach. So we have this library spectrum, right? We've heard about this all week already, and these major ions here that we see match these kind of colored guys which are buried in the SWATH spectrum. And also to remind you that what we're doing here is this extracted ion chromatogram based on these fragmentines. So once we focus in on these fragmentines, it becomes much cleaner. You've also seen this picture several times this week. This is the assay library. Except now we change it a little bit. We don't really care anything about instrument operation parameters. These are not required in the library, so we can kick this out because we're doing this. This library is being used post-acquisition for analysis, right? So now it just looks like this. But in principle, the library has this very similar set of information: the protein accession, the peptide sequences, the precursor and fragmentine, M over Z's, some other information, which ion type it is. Importantly, the relative fragmentine intensities and the peptide retention time on some normalized scale, this IRT scale. And this is essentially the starting point for this targeted analysis, right? Some picture of what the peptide looks like to the mass spectrometer. And so just to kind of walk through this targeted analysis concept one more time, just to reinforce this, what we're starting with here is some DIA data, some SWATH data, and here's again this complex multiplexed MS2 spectrum where all these fragmentines are arising from many different precursors. But now we're going to say we're going to do a targeted analysis. We're going to say, okay, I want to query for a particular peptide. So here's some peptide sequence that I already know something about. I've already observed it in DDA mode. And I know that these are the major fragmentines for this peptide. So we can zoom in and say, oh, okay, this is the Wi-Fi fragmentine. I know it should be at this mass, and here's a nice isotopic envelope. This is the Wi-Fi fragmentine. And when we integrate this information over time, if we extract all these ions, XIC extracted ion chromatogram, as Jared described, then we get something that looks kind of like pseudo SRM data, but and essentially we can now feed this to the M-profit approach. So this is what Ludovic explained in a lot of detail this morning. It's conceptually, I mean, very, very similar. We have some additional scores that we can calculate. I'll mention a few of these, but this is essentially what I mean by the targeted data analysis workflow. So this was kind of first came out of our lab, this paper from Ludovic in 2012, and yeah, just to encapsulate the kind of idea of this paper is we're going to do this windowed acquisition, data independent acquisition. We're going to build in some prior knowledge in terms of these SWATH coordinates, it's called here, but this is basically the spectral library or the assay library, and we're going to end up with this chromatographic quantitative data. I just took one figure out of this paper, one kind of key figure. Somebody asked in the previous talk, you know, how do these different techniques compare, and this is something that Ludovic looked at the time. So these little circles are the DDA identification, whether you could get an identification from this peptide in a DDA run. The black triangles are the SWATH. So I should say this is a dilution series, right, of this particular peptide into some background. So we're going from 100 femtomoles down to less, down to 10 attomoles. And the signal from this black triangles is the SWATH signal. The signal from the little x's is SRM, and the signal from the white squares is MS1. So essentially what we can try to define is the limit of detection for the shotgun identification is at a given concentration. The limit of detection for the MS1 label free is at another concentration, a bit lower down. The limit of detection for SWATH is a bit lower down again. And finally the most sensitive was SRM. And this was an experiment that was done at the time, but I think we have other data sets which kind of broadly agree with this conclusion. The limit of detection for SWATH is somewhere in between SRM and MS1. And so now the software tool that we use in our group, that was developed in our group to do this targeted data analysis, is called OpenSWATH. So this is written in this OpenMS framework, C++. It's all open source and using open data formats. And this flow diagram here is essentially exactly what I've showed you already, right? So it encapsulates this targeted workflow, but it's now kind of formalized in terms of the software. So we need two things. We need the SWATH data, which comes in the form of a MZML file. We need the spectral library, which comes in the form of a TRAML file, another XML file container for spectral libraries. We do one step of retention time alignment where we essentially take the spectral library assays, which are stored in IRT space, and we project them onto the retention time space of the run, so we know accurately where the assay should appear. Then we do extracted ion chromatograms, and this is depicted here, against whatever library you choose to provide to the software. It could be big or small. We do feature scoring. This is exactly what Ludovic described this morning, except he described it in SRM terms. I'll mention a little bit more detail about the scores. And then a statistical analysis based on M-profit, based on the M-profit idea, also exactly as Ludovic described this morning. So a little bit more about the scores. So these are more or less the scores that Ludovic told you about this morning that are calculated based on the chromatography. based on the extracted ion chromatograms. And this is what he told you. You know, you can check how far away it is from the expected retention time, do all the transitions, co-elute, is the shape similar, is there an intensity, is the intensity higher generally than the noise, are they kind of Gaussian shaped peaks, or you can also calculate a signal to noise ratio, and also the relative ion intensity with reference to the MS-MS spectrum. In SWATH data, however, we now have this full scan MS2 spectrum, which is high resolution, so we have additional, I think there was a question about this earlier, we have additional scores that we can compute. So in addition to, say, like the 5 or 6 transitions that we chose to extract from the data, we can look back in the MS-MS spectrum and see whether the fragment ions for the rest of the peptide ion series is there, right? This is additional confirmatory information. We can check the mass accuracy of this peak, you know, each one of the fragment ions, is it close to what we expect it to be based on the theoretical calculation. We can see if this isotope distribution is close to what it should be, what we expect it to be for a fragment of this mass, and we can also check whether whether this is the monoisotopic peak or whether there's interfering isotopic overlap. So there's many more scores that you can compute for scoring the SWATH data as compared to SRM, because we have this full scan MS2 high resolution information. And I just want to point out that, so you already now did this tutorial, so you recognize all these scores, right? And these are basically the same set of scores that are included in Skyline. You did it for SRM data, so you kind of mostly had these scores, but some of these grayed out ones that you probably couldn't select relate to this high resolution MS2 data. So when this software, OpenSWATH software was developed, the developers needed a data set in order to kind of benchmark this and to say whether it's working or not. So in order to have a sort of a ground truth, what they did was they went around the lab, gathered up as many aquapeptides as they could find, which turned out to be about 400. And they pooled these together and did a 10-step dilution series from 50 femtomole down. And they put these into water, yeast, and human backgrounds. And then they measured this in SWATH mode on the 5600 QTOF instrument, and they got a whole bunch of data. And then what they did was 3 or 4 of them sat for several days, and they manually inspected all the chromatography in Skyline, and they called it, this is a peak, this is not a peak, right? So they went down the dilution series saying when they had the correct peak or not. So now they have a kind of a ground truth. They have a set of true positives that they can use to check whether their software is working. And this required a lot of, they were kind of grumbling because they had to inspect so many chromatograms, but out of these 30,000 chromatograms which were extracted from the data, about 19,000 true peaks were annotated, and this was used to check whether the software is working essentially. So here's the result of that comparison. So this is a sort of this is a receiver operator characteristic curve. So sensitivity means when the manual analysis said there was a peak, how often could the software also detect that peak? And what they get up to is about 90%. And in fact, in the manual analysis, the peak groups were somewhat over-annotated. We think so. As you go down the dilution curve, right, it's always very easy to annotate the same peak every time. So this means that this missing sort of 5 or missing 10% in a real manual inspection you might not be able to find, but because it was a dilution series, it was easy to find these peaks. So even this is kind of a little bit unfair against the software. But the point is that we can recover the signal in kind of 90% of the time. And then the second question is, I told you that OpenSWATH is also doing the M-profit style approach to estimate the false discovery rate. And so they could also check against the manual analysis, did the software perform, and was the false discovery rate that it estimated consistent with what was known from the kind of gold standard truth. And this is the true FDR as determined by the manual analysis, and this is the estimated FDR, and you can see that it basically is in more or less in good agreement. So, oh yeah, I wanted to say that this point also goes to what Jared said in terms of data completeness. So, and the idea that we're trying to detect peptides here. So what we're trying to achieve is something which resembles an operator inspecting manually the data in Skyline. So if the peptide is there, you should find it. If it's not, then you shouldn't find. Then it's not there. But you don't have to rely on any data-dependent acquisition. And this kind of data completeness concept is an important one that you've heard about all week in targeted proteomics. But this is what they were trying to essentially recapitulate. That if the signal is there, then the software should find it. If it isn't, then it doesn't. They also checked whether the quantitative performance looked kind of reasonable compared with these standard peptides, and the short answer is that it is. So this is the manual analysis in Skyline. This is the automatic analysis in OpenSWATH, and I mean it's basically the same. So the quantitative analysis also was recapitulated. So if you were to run an OpenSWATH analysis, what would this kind of output look like? So essentially, after you go through this whole workflow, it returns a table which looks like this. So you have sample number 1, sample number 2, sample number 3. Here's a particular peptide at a given charge state, and it will report a sum, the peak area sum, or it will report the fragment ion peak areas for these individual fragments, and importantly, this Q-value score. So the estimate of whether this peptide is actually really what you think it is. So, and this is derived from this M-profit type analysis that was already explained. So these two guys have very low Q-value scores, so we say these are true. This one has a big Q-value, so it's probably wrong. Yeah, and there's the peak areas. And what you would do downstream of this, so what we get from OpenSWATH is this fragment ion level, peak areas, and also a call of whether this is a true or false peak group. But for the protein inference and statistical analysis, you still have to do something downstream. So you will hear, and there will be a tutorial on this MS Stats software, which is implemented inside of Skyline now. But these are some of the tools that we commonly use. But just to point out that OpenSWATH is stopping at the point where you sort of detect the peptides and quantify them, and this assay level or peptide level error model, but it still needs further processing. So this is more or less the summary of the first part. So what you need is some SWATH or other kind of DIA data, similar to what Jared described. You need to have an assay library, which we've heard about all week, how to derive these. And actually, there's another talk tomorrow which I'll explain in more detail exactly how we get to this prior knowledge from the DDA data. And the analysis is based on these XIC, extracted ion chromatograms. The M-profit scoring tells you whether you pick the correct peak group or not, but also some false discovery rate control, and the output is this quantitative fragment level data. So is there any questions to the first part? Okay, good. So now I want to look briefly in this kind of a few applications. And I've listed what they are, and the point is not to tell you anything about the biology of these particular applications. I just thought it's interesting to kind of look at the different scales of which this type of analysis can be done. So the first one is actually in this original SWATH paper from Ludovic. They looked at some yeast data in a classical yeast experiment where basically the yeast are fed glucose, and then they eat all the glucose, and then they have ethanol to eat. So it's a metabolic change in the metabolic pathways of the yeast. This is a paper we worked on a couple of years ago where we were looking at interaction proteomics and protein complexes, which a number of people in the group were interested in. And this is now, so this complexity, by the way, is not really, I'll explain afterwards. This complexity is not the complexity of the organism, but more the complexity of the analysis, and rather the number of proteins we tried to quantitatively extract. But the biggest one is this one, which Rudy already mentioned earlier in the week, where we sent samples around to many different labs, and we basically tried to identify as many proteins as we could, and there's a lot of data files, and it's a very large-scale analysis. Okay, so let's look at the yeast one first. So this was the first paper, and essentially what they did was they picked kind of, and actually this particular project had been studied by SRM before and also published on with respect to SRM. So what they did was picked kind of 30 or so proteins which are in the glycolysis pathway, and checked how their regulation changed with respect to this particular perturbation. And they also correlated this with so they took these 30 proteins, 40 proteins. They had the spectral library, and they extracted the quantitative data for these proteins from 2 files. And basically, these colors are the up and down, fold regulation. Something is happening in the glycolysis pathway due to this experiment. So this is a relatively, I would say, SRM-style focused hypothesis-driven analysis. And it agreed very well with the SRM-type data. They then said, and this point also came up, was in Jared's session during the questions, he pointed out that this type of iterative data analysis is possible. So after the fact, after the measurement, somebody said, well, maybe this electron transport chain is affected as well by this particular perturbation, and then they went ahead and re-extracted data for another 36 proteins. And this essentially, but again, another sort of hypothesis-driven and small-scale analysis. But it highlights the idea that you can do this sort of iterated data analysis. The second example is this affinity purification study. The details are not so important. I mean, I will quickly say that there was some cells which had a particular perturbation, a growth factor treatment, and we purified this 1433 scaffold protein under different conditions of the growth factor treatment to check how the composition of this protein complexes were changing. And from this we got a very complete data matrix, and we could say something about what the protein interaction network looked like, but also something about the dynamics. And for this we had, it was sort of a relatively focused sub-proteome experiment, but we had no particular list of targets that we were interested in quantifying. We just kind of wanted to see what we could see. So we generated the spectral library internally in the experiment, and we could, by DDA, and then we took that same library and the same samples shot in SWATH mode, and we used that library to extract the quantitative data. And from this, we could extract about, in the SWATH files, we could extract about 2,000 proteins from about 2,500 that were in the library. So I should say these numbers, by the way, are on the right is the library size, and this is the number that we could actually detect. So of the 30,000 peptides in the library, we could detect about half of these in any single run. In the protein level of the 2,500 proteins, we could detect about 2,000. This was a little bit bigger study. We went up to 27 SWATH files, but still a kind of moderately manageable experiment. And then the larger one is the one that Rudy already mentioned earlier in the week, that we were interested to look at the performance of this method across different sites. So we sent out this HEC293 lysate to 11 different labs, and we asked them to run it all basically 20 times. And the point Rudy was trying to make is that you detect a similar set of proteins across all the sites, which is nice. The point I would like to make, the library that we used for this was a very large library. So I'm going to say more about it tomorrow, but this library that we used is essentially consists of the DDA data for every human sample that we have in the lab. We aggregated it together. So there are about 10,000 proteins in this library and about 140,000 peptides. And we don't expect most of them to be in these samples. So we detect 30,000 out of this 140,000 peptides. We detect 4,400 out of the 10,000 proteins. And this is done over 220 SWATH files. And so I guess what I'm trying to point out, I will say more about this. Question came up earlier, but I'll say more about this tomorrow. Special kind of considerations with respect to the false discovery rate are definitely required when you go to these very large scale libraries, and especially Brendan already mentioned prevalence earlier in the week. So if the number of peptides you can actually detect is much smaller than the number of the library, this has implications for this false discovery rate. But I'm going to defer this until tomorrow when we talk about how to build libraries. So this is kind of the summary of these 3 applications, right? So we have this yeast, very kind of hypothesis-driven SRM-style quantification with known targets. We can do this iterated analysis. We measure, in the end, it was about 105 proteins, and this is a very focused study, right? So this is definitely targeted proteomics. So my question here is is SWATH still targeted proteomics? The interaction proteome study, we measured about 2,000 proteins across 20-something samples. We didn't know what we wanted to measure in advance, but it was still, in biological terms, a kind of a focused study. And then the third one is really the kind of true discovery type of study where we say, we're just going to try and quantify whatever we possibly can and see whether it works or not. So there's no hypothesis whatsoever. We're just going to measure whatever we can, but we're going to try and measure it in a consistent way. And the question is whether this is still targeted proteomics or not. I mean, I don't really care. The point I'm trying to make is that we still apply this targeted data analysis paradigm, and this is what leads you to these kind of matrices that have very complete values. This is what Bruno showed before, what Jared showed before, that by having this fixed duty cycle and having this targeted data analysis, we can get to very complete data matrices. So we can do it in a small scale way, we can do iterated analysis and re-extract things, or we can do it in a very large scale way and try to find out whatever we can. So the sort of semantics of whether it's targeted or not, I mean, it's definitely not hypothesis driven, but it's a targeted data analysis, and it's our goal is to keep data completeness. This is the part that I'm more interested in. Okay, so this is more or less what I wanted to say about these kind of more targeted data analysis methods, and I'll mention tomorrow in a bit more detail where these libraries come from and so on. So we now already talked a little bit about this in the last session. I just wanted to pick out one of these kind of more untargeted methods and show a couple of slides on this, just because it's becoming popular again. It's kind of an old idea, as Brendan mentioned, came from this Waters MSE idea, and it's now been re-implemented by Alexei Nezvishki in terms of this DIA empire. So the way I would explain this is that here we are back to our very complex MS2 spectrum, and I told you that there's this peptide in here that has these particular fragment ions, but we can't search it with the mascot, as was already said. So the way we deal with it is to build in the prior knowledge of the spectral library and extract just these things. But the question is whether there's another way to deal with it. And this is what Brendan already mentioned, this MSE idea. So, and they do it, actually, it's even harder challenge for them because they don't even do precursor window isolation, they just fragment everything in the MS1 range. So here's 3 peptides which you can observe by MS1. This is the fragment ion spectra when you co-fragment all these 3 together, and here we are again with this complicated spectra that we find difficult to interpret. Is there some way to deconvolute this? So what you can do is do a feature finding step on both on the MS1 and MS2 data, and then extract ion chromatograms on whatever you find in the feature finding step, right? So here's the XICs of these 3 MS1 precursor ions, and they co-elute, but they have a slightly different elution pattern. Here's the XIC at the MS2 level of all these fragment ion data, right? So I've colored them in so you know what they are, but at the start we don't know what goes with what. But by correlating whether all these MS2 fragment ions correlate with each other in the retention time space and also with the precursor in the MS1 space, you can sort of link the yellow fragment ions to the yellow precursor. They have the same elution pattern, and link the purple ones to the purple and the red to the red, and now you have deconvolved this very complex spectra into something that's more manageable and potentially manageable by a standard DDA search, right? Because they call this like a pseudo MS spectrum, because you have now a precursor, and what you expect is that this set of fragments belong to this precursor, and that's all you need to do a standard DDA search in mascot or something. And this was kind of the Waters implementation. Alexei has kind of implemented this idea in this DIA empire software. So the workflow here is essentially what I just described, the signal extraction. So where you take the DIA data and you try to group together the precursors with each other, but also, sorry, the fragments with each other, but also with the precursor. And then you do a regular DDA search. And somebody asked, can you after you do this deconvolution, can you put it into mascot? And the answer is yes, you can put it basically into any search engine you want to. Alexei and these guys tend to use this transproteomic pipeline tools, but we can now query this against the sequence database as we would with any normal DDA file. And so now you get this set of identifications, and if you want, you could do try to do the quantification at MS1 or MS2 level. But what they found is more effective is to say, okay, now we have a spectral library. Now we're back to the targeted approach, right? We have a spectral library, but we've generated it internally on the SWATH data, so we never had to do any DDA analysis. And essentially, we're now back at the point of everything I've just described before this, that you can do a targeted extraction. And now what they say they get from, and they do something kind of similar to what OpenSWATH would do, and what they have now is very high coverage quantification. So they went from quantitation to high coverage quantification over here. So essentially, they're trying to get to this very complete matrix that we said we already have from the targeted data extraction. And so oh yeah, they also mentioned that this is now compatible with external spectral libraries, so you can do kind of a hybrid approach. You can generate your own library internally, or you can download spectral libraries and do this targeted extraction based on both of those. This is a few results from Alexei's paper. They did a direct comparison of DIA umpire with OpenSWATH or with DDA. So this is at the peptide ion level, or at the protein level. And actually, in their comparison, so this is their comparison, they found DIA umpire performed very well, a little bit better than OpenSWATH, and the DDA was kind of somewhere in the middle. At the protein level, at the peptide level, the protein level was very similar actually. But I mean, we actually talked to Alexei a lot about this, and we have some quibbles with the way they ran OpenSWATH. But anyway, the point is that it seems like you can get to a more or less similar results, at least in the data that they tested, by going for this untargeted approach. So this is quite new. I just wanted to mention it because, you know, they sort of are saying that this is the future, some kind of hybrid approach where you take external libraries and internally generated spectral libraries and try to get everything that you can. I think it's an interesting idea. Okay, so now we're basically at the summary. So what I would say is that this kind of targeted data analysis based on assay libraries is what has made SWATH or DIA work, at least in our hands. Inputting this prior knowledge allows us to kind of sensibly handle this highly multiplexed MS2 data, MS2 spectra that we generate in SWATH mode. The XIC-based analysis combined with DIA facilitates a very high level of data completeness, and this is really what we want to maintain, right? We're trying to maintain the same level of data completeness as has been achieved with SRM and so on. And that this targeted data analysis approach scales from this kind of SRM-like, you know, 10 or 100 proteins as typified in the original SWATH paper. We're now trying to get this up to thousands of proteins, which we can achieve in OpenSWATH. And actually, I've been talking with Brendan this week. He's done a lot of optimization to make it possible to do these large library extractions in Skyline, which is great. And yeah, again, the idea is to try and maintain this data quality across this scale. I would say in this, again, the caveat again is in these very large extractions where you have very large libraries and low prevalence and large numbers of files, there are definitely other FDR considerations to discuss other than the ones what Ludovic mentioned today, but I'm going to come back to this tomorrow. And now finally, this kind of untargeted methods and many people are doing different things in this area, are coming online and seem to be becoming competitive with the targeted analysis approaches. So this opens up the possibility of kind of making hybrid methods from these different approaches. Okay, so I'm going to leave it there. I'll try to. I think I went through a little faster than I wanted, but I'll try to take any questions if anybody has one. Yep.
Just a sec.
Oh, right, sorry. Yeah, the microphone.
For this data deconvolution, are you aware of any software at the moment, except PLGS, of course, which is capable of including this drift time or eye mobility data for spectrum?
Good question. The short answer is no, but I do know that Alexei went, that I don't know, sorry. No, I don't know. So I know that Alexei went to a lot of effort to make DIA umpire compatible with different data types, including MSX, and they've analyzed fusion data and other DIA type approaches, also this P-Smart thing from Thermo, or various different things they went, but I don't know if he did it for the waters data or not. So that would be a good question for him. The paper just came out, and like, for example, they're still developing it. So this external library stuff wasn't in the first version. It just came in a couple of months ago. So yeah, right to Alexei.
Thank you. May I please ask, so in principle, if 2, 3, or 4 different labs perform out the same experiment, then the data sets should be fairly identical. They
That's actually what we're trying to figure out. But
I'm just talking about a hypothetical
sure.
situation. So how does that work in reality? Because I think one issue that also was discussed earlier is when people carry out similar or identical experiments, the outcome can be very different in proteomics. So would SWATH be able to resolve this problem?
I mean, that's one of the big hopes, right? So I would start by saying one of the reasons that drove people in the direction of SRM and kind of smaller scale targeted proteomics is what question you asked. Can you kind of do the same experiment across labs and get robust values? And this was the purpose of this CPTAC consortium in the States that Brendan mentioned was originally funding Skyline. They were sending around these standardized samples to different labs and checking if you do SRM measurements on these, do you get basically the same answer? And they published a couple of papers on this, and I mean, the answer is that like the sensitivity was a bit different in different labs. The quantitative characteristics was a little bit different in different labs due to different setups, but broadly the answer was the same, I would say. And this is also the point of this study right here. So this was Rudy's idea. He figured that there was enough people doing SWATH now, about a year ago this was, that we could send some samples around to these 11 different labs. They would all acquire the data on exactly the same samples, and then we would do the data analysis and check, do we get the same answer? And this is one plot showing that, well, it's not identical, but it's kind of not too bad actually as well. And there's Rudy showed some other quantitative data saying that, you know, in these aquapeptide dilutions, you get to a first approximation, you get the same answer or a similar answer. And this stuff about what you mentioned about reproducibility of research is exactly what we'll sort of write in the introduction to this paper, I hope. Does that kind of answer?
Yeah, thank you. May I just add,
of course,
in a future perspective, I think this is now absolutely great, and I think all the people who tested it, this is the world top elite, but for all the B-level labs, to put it like this, who do a lot of work and useful and important work, would that work too?
Very good question. There is a couple of core facilities involved in this study, not all, but I think there's maybe 2 core facilities. It's true that most of them are kind of research mass spectrometry labs. Something I didn't mention about this study as well is that we had a sort of a QC round. So we sort of, before we did this measurement, we sent around a representative sample to everybody and we said, just shoot it a couple of times and see how it goes. And there the data looked a bit more heterogeneous. And so what we decided was to write in a bit more detail a protocol and sort of help people to quality control their data, right? So, oh, the peak width is a bit too wide, do something with your chromatography, your signal is degrading, clean the instrument, stuff like this. So I think if you document these procedures, like what to do in case the data isn't working, your data doesn't look good, then it allows people to fix it, then I think most labs will be able to follow these types of procedures. I don't know if we really have completely the answer to that question, but there is 1 or 2 labs in here which were core facilities, and they also did okay. Another thing I would point out, though, is that all this data was analyzed centrally, mostly by me, actually, and if we then did the data analysis, let everyone do the data analysis locally, I think you would run into probably more problems. There would probably be more heterogeneity if we said to everybody, just analyze the data and do the best you can, then it would look pretty heterogeneous, and I think you would need to standardize that data analysis as well. But you know, the first iteration of this was to just try and do the measurements everywhere and see how that impacts things. So I think that your question is good. I don't think I answered it fully. I answered as best as I could, but that's the state of, that's as much as we can say about it now, but I want to point out that we are thinking about this and other people are thinking about it.
Yep. Thank you so much.
You're welcome.
I just try to formalize the question. How difficult is it from your experience to use spectral libraries from repositories? They are created in a specific matrix and you want to measure in another matrix.
Right. So that this is covered a little bit in tomorrow's talk. I guess I will make a short answer now. We haven't looked that much into the matrix effects, I have to say. We've looked a little bit more into the instrument variation and what impact this have, whether you measure the spectral library on one instrument or another. The matrix effects, my guess is that, and this is a bit speculative, but imagine the case that you're now going to make large-scale spectral libraries and you start to aggregate peptide spectrum matches that came from different samples, right? One came from a tissue sample, one came from a cell line or something. And when you aggregate them all together, what we do is we make a consensus spectrum out of these. So we essentially kind of average them out. And there's a specific software that we use for this called Spectrust, and it does the consensus of these spectra in a smart way. So there's some quality control involved. There's kind of a majority voting to see which peaks you keep, which ones you throw away. Stuff that's a complete outlier, if there's one peptide spectrum match that's a complete outlier, it'll just trash it, right? And so the more data that you aggregate on this and average over, the more I would hope that these matrix effects would go away. Second part is the kind of instrumentation, the mass spectrometer fragmentation and the retention time and stuff. And I can say that in this study, we used one spectral library, right? But the data was collected at many different instruments. So they're all the same instrument, they're all 5600s, but I mean, I think we can conclude from this that it basically works at least across the same instrument. We have looked into differences in fragmentine spectra across different instrument types, and this has already been discussed a bit. I will show one or two slides about this tomorrow. But what I would say about that is that it does get worse as you get further away, right? And the HCD instruments look more like each other. The beam type collision instruments look more like each other than the CID instruments, the sort of trapping CID instruments. The question is whether this actually, you can still use an Orbitrap HCD spectrum to query against swath data on a 5600. So it may not be perfect, but it might still contain useful information. It might be close enough. And you saw that in this MProfit approach described by Ludovic either, it's learning on the fly what are the best scores. So it may be that, you know, all the rest of the scores are fine. This fragmentine spectrum is a bit worse, but maybe the machine learning downweights this a little bit and maybe it's not as crucial. So in practical terms, we haven't done so much evaluation on this, but I would say that it's not a black or white, right? There's a continuous, so you should try to get the spectra as close as possible, but maybe the next instrument over might also work too. This needs to be looked into more. Ralph?
Just a very brief question about the 5600. You mentioned you have 25 M/C isolation windows. Is this the way you run the 56, or do you normally?
Not anymore.
So how do you run it?
So the original paper, it was 25 MOZ times 32 windows. And somebody mentioned earlier that AB Sykes has this variable window setup where you basically size the windows based on the density of the signal. So now in the kind of, you know, I forget, like in the lower mass range around 500 or something, 500, 600, the windows are like 8 M/Z wide. And up at 1100, they're almost 100 wide. And there's 64 of these windows in our standard method. So we went from this 32 times 25, 32 windows times 25 M/Z units. And now we have 64 windows in our standard method, and it varies from 8 up to 90 or something like this.
And this is implemented in the latest analyst version? Probably, huh?
So you would need to speak to AB about this. I don't know that it's in the sort of off-the-shelf version of Analyst 1.7, but if you ask Christy nicely, she'll help you out.
Okay, thanks.
Now I said that on video, so she has to. Other questions?
Since you are fragmenting everything coming in the mass spec,
Yep.
For sure, the energy you are applying will reflect what you will obtain on your peaks, okay? And how do you manage with the energy? Because if you change parameters, for sure you will not have the same thing. And even when you are trying to quantify, you will not have the high, the
So you mean
intensity.
you're talking about collision energy?
Yeah.
Okay, so that's a very good question. There's also something I was going to mention tomorrow, but I can quickly summarize that so what Jared said is that you cannot optimize the energy for all the peptides in the window. So we do the same thing as him. We set it for a 2+ ion that's kind of in the middle of that window. So for the swath analysis, it's each window has a fixed collision energy across the whole run. You know, we can't make any on-the-fly decisions about this. Something you can do, though, is when you're getting your spectral library, you're doing the DDA runs, you can adjust the collision energy in the DDA analysis to match what it would be in the swath analysis, right? So even if you have, so you can set the instrument up to say that, you know, usually the collision energy is calculated based on the charge state and the mass, right? But you can say, well, even if I have a 3+ ion, I'm going to compute the energy as a 2+ ion, which may not be optimal for identifications, but it does make that spectrum look more similar to the swath spectrum. So maybe we sacrifice a little bit of identifications, but we gain fidelity in the matching between the assay library and the swath data. So in general, and it's not only for collision energy, but other parameters, you know, you want to try and get things as similar as possible, and then this is the most effective way to get this targeted analysis and scoring to work. A bit more on this tomorrow. Yep.
Thank you. May I ask one more question? Now, we have looked mostly on expression of proteins, but some proteins have very complicated modifications. Well, we have covered that briefly, but if you take now, let's say, terrible sample such as histones, what is your view about this kind of samples?
I guess I can't speak specifically about histones. I mean, I can speak a bit more generically about post-translationally modified peptides. So these are more challenging. Actually, Ariel is going to talk about phosphoproteomics tomorrow, probably. No, Friday. So he will go in a lot of detail on this targeted proteomics, targeted phosphoproteomics. It is more tricky. There's certain things you have to be careful about, but in principle, if you have a library spectrum for the post-translationally modified sample, then you should be able to extract that signal in the swath analysis, right? And you may need to be careful about certain things, like if you have a peptide which is post-translationally modified at one location, at one amino acid in the peptide, or there's also another possible isoform where it's modified at another amino acid residue in this peptide, then sometimes the signal in swath for these two peptides can look a lot like each other, and you need to be kind of careful that your library spectrum doesn't kind of cross-match from one to the other. But in principle, there's nothing stopping you analyzing post-translationally modified peptides, but maybe there's some more precautions you need to put in place in terms of the spectral libraries and how you interpret. So especially the sort of how you interpret the chromatography. So usually these two isoforms will not be overlaid in the chromatographic space, right? So because we have very complete data over the chromatographic time dimension, usually these types of peptides can be resolved in the chromatographic space. And you just need to be careful that your library, if you have both versions in your library, that it points to the right one and not the other. Did I explain myself, or?
Yeah, sure. Thank you.
Anyway, more about PTMs from Ariel on Friday. Okay, so if that's it, I guess we will finish up, and I don't know what's next.
Skyjam.
Skyjam, directly into Skyjam. Great. Lucky you guys.